All right, now we are at our third chapter, which is sensation and perception. And I'm going to do my best to just bust through this one so we don't have another hour-long PowerPoint. So here's a quick overview. So here are the basics of sensation and perception. Sensation is basically what is coming in. Perception is basically how we interpret what came in. And then, of course, here are the actual definitions. So these are good to know, but just understand sensation is what's coming in, perception is how we interpret it. All right, how do we measure sensation and perception? Absolute threshold is the minimum intensity of a stimulus you can detect half the time. This is different for everyone, hence the use of the 50% marker. A just noticeable difference, or J&D, is the minimum change of intensity of stimulus detectable half the time. So in Weber's law, Mr. Weber believed that just noticeable difference, or J&D, depends upon intensity of stimulus when first presented. So think about it like loud sound at a concert. The sound changes depending on who you are, how many concerts you've been to, and how long you've been there. So this law aids us in understanding the subjectivity and why we have that 50% mark. Subliminal perception is unconscious perception. So it's like the brain taking something in without being aware of it or paying attention to it purposely. So think about all of the self-help tapes, and there's scenes in a billion TV shows where somebody is like gonna sleep with a self-help tape going in the background, and they use it to either, you know, stop eating or stop smoking or be more aware, things like that. There are no studies that support this being a viable method of persuasion, although advertising still tries. Even though it's not viable, it can be effective-ish in advertising. But a photo or a series of photos are not going to brainwash us into doing anything or buying a product. But if you connect with some images or some themes, they might affect our attitudes, or they might appeal to our beliefs, which then makes us want to buy something or use a product. Again, self-help, there are no studies shown to support that they make any sort of difference. And it's all about if you connect with a picture or an image, you might want to buy it no matter what. ESP, which stands for extrasensory perception. This is the ability to acquire information without using your senses. So it is basically reading people's minds and being able to tell the future. There's absolutely zero research to support that this exists. Zero. They've used all sorts of experiments and brain scans and all sorts of things, but nothing ever worked. Okay, remember all the important stuff in red. So sensation is the process by which sense organs convert input into neural impulses, and perception is our interpretation of that information. So again, this talks about subliminal advertising, not really thing. However, I will always fall for the coffee pictures. They make me want coffee so bad. There's another quote-unquote subliminal image. And yet another. All right, now we're going to talk about vision. When you see an object, you are looking at the light reflecting off of it. So light is electromagnetic energy. Your eye converts that energy on a visible spectrum. So different animals can see different parts of the spectrum beyond our limits. Like, you know, dogs and cats can see better in the nights than birds are okay with UV rays beaming into their eyes, etc. What we see is defined by the characteristics of the light we see. So wavelength is distance between peaks of consecutive waves. They correspond with colors. The shorter wavelengths are cool colors like blues and purples. The longer ones are warm colors like yellow and red. Amplitude is the height of the peaks, and that corresponds with the brightness of light. The higher amplitude is brighter, and that also means brighter color. Also, saturation equals the purity of light. Here's a nice, lovely little visual for you. So you could see what we mean by the peaks, the very high peaks, and then also the wavelengths. All right, let's learn about the anatomy of the eye. Here we have a lovely diagram of the eye that is also in the book. Let's start with the cornea. The cornea is the clear, bulging outer surface of the eye. It protects your eye and starts the focusing process. The cornea's shape and damage play into how well a person can see. And contact lenses, and to some extent glasses also, correct for this by being thicker or thinner in certain places to shift the way the light hits the retina. The pupil is directly behind the cornea, and it's like an aperture where the light passes through. Because the light can't go through the sclera. And the sclera is not shown, but basically it's the white part of the eye. All right, and then we have the iris, which is the colored part of the eye. And it is controlled by a ring of muscles that control the size of the pupil, which in turn controls how much light is getting in. Then we have the lens, which is behind the iris and the pupil. It's a clear structure attached to the eye. It works like the lens of a camera. It brings in light and focuses by changing shape. And that's called accommodation, which is very important. So please make sure you're writing this stuff down. The retina is the surface lining the back of the eyeball insides. It contains rods and cones, which are specialized cells to convert light into neural signals to travel down optic nerve. Here they are, rods and cones. Ooh. The signal goes back to rods and cones, which convert the light, then get sent through horizontal, bipolar, and amacrine cells to the ganglion cells on the retinal surface to the optic nerve. The optic disc, which is where the optic nerve leaves the eye, is a blind spot. There are absolutely no cones or rods there. It is then compensated for by your other eye, so you never even notice it. So rods and cones. Rods are the long and skinny ones. They're black and white receptors. Cones are shorter and fatter, and they transmit the color information. So here's a good memory trick. CO cones equals CO colors. Cones need more light to activate. That's why we see shapes when it's dim, but not always the colors. Think about driving in the dark. You see cars, but you can't always tell what color they are. You can just see the shapes of them. So at the fovea, which is the point of highest visual acuity, the cones are concentrated here. So more rods at fovea, and then there's less as you move out. Photopigments are chemicals activated by light. When the light hits, photopigments break apart and sends a chain of chemical reactions that send a signal, and it has to do with what we already know about neurons and action potentials already. All right, here we have the lens. And remember, I told you to pay attention to this part. Accommodation is what we're going to write on the back side. And please also know what accommodation means in terms of the lens. All right, let's talk about light, dark, and color. Dark adaptation is a process the eye goes through when going from light environments to dark. And then obviously light adaptation is the process when the eye goes through when going from dark to light. Remember the photopigments that we were talking about? They split up with the light. So no light equals no photopigment breakage, which causes a buildup in the eye, so when the light comes on, it seems way brighter than it should, or might even look like a flash and could be painful. Way more photopigments are in there to receive that energy. And vice versa, when going from light to dark, the pigments don't have as many available for when entering the dark. They have to have a buildup to see. So the eye has only one kind of rod, but 3 kinds of cone. Remember this. It ties into the next theories. So 3 kinds of cone, and there are different photopigments in each. We see 3 colors with 3 cones. Do you see where this is going? So there is the trichromatic theory of color vision, which is the idea that color vision is possible by presence of the 3 types of cones that react to either of those 3 colors. So think of it like paint, basically. You're mixing colors, and each time you mix, it releases certain proportions of each type of photopigment. So this doesn't explain after image, which is when you stare at something in one color, like red, and then look on white, and then it's green. So maybe you'll remember the dress theory that was on the internet a little while ago about, is the dress gray and green or is it pink and white? That has to do with the after image. So opponent process theory of color proposes that we have dual action cells that signal the brain when we see pairs of colors. So for example, red and green, yellow and blue, black and white. It's called opponent because you can only see one or the other at a time. This explains the after image in the same way as the light-dark adaptation and use of photopigments breaking up. Too many or not enough. So which is right? Which theory is correct? Many think it's both. They are processes that operate at different places within the visual system. So trichromatic at rod cone level, and the opponent beyond that level, perhaps in the ganglion cells or even in the brain. Color blindness is usually the result of missing cones in the retina. So the most common is red-green blindness. You're either missing the red or the green. This occurs in 8% of males in the US, and it is linked genetically because women rarely have it, and other countries have different rates of color blindness, enough to be statistically significant. So here's the test. All right, so here we have this lovely shades of red and green dots. And if you have either red or green color blindness, all you'll see is a whole bunch of dots. If you have good vision in terms of color, then you should be able to see an image in the dots. So in this case, it is a number. So everybody can try it out and report back. All right, moving on to hearing. Just like light, sound travels in waves with peaks and valleys, and there's information on each peak and valley. Each peak and valley is a cycle. The number of cycles, which is one peak and one valley, in a given time determines frequency, which is measured in hertz or Hz. So it's measured in hertz number of cycles completed per second. And of course, high frequency equals higher pitch, and low frequency equals lower pitch. And of course, I feel like we already know this, but hearing loss comes with age and from working in noisy environments. All right, the anatomy of the ear. We're going to start with the pinna, which is the outside of the ear, or as we like to call it, the ear and earlobe. It acts as a funnel to gather sound. So think about dogs. They can move their ears and change shape to hear, just like putting a funnel in your ear will help you hear better. The auditory canal makes sounds amplified and strike membranes at the end called the eardrum, which is a thin membrane vibrating as sound waves hit it. The middle ear bones amplify the vibrations and send them on to the inner ear, where there is, you guys can see in this red square, a hammer, anvil, stirrup. So I guess you could, as a memory trick, think of an Old West town with a blacksmith who rides a horse from the middle ear to the inner ear. The stirrup rests against the oval window connecting the middle ear to the inner ear. The oval window is on the outer edge of the cochlea, which is this purple snail-looking thing that's filled with fluid. And this is where vibrations get turned into neural signals and travel through the auditory nerve into the brain. The inside cochlea is the cochlear duct, whose floor is lined with basilar membrane that has hair cells that do the converting. This happens because vibrations in the middle ear make the stirrup hit the oval window. So again, with the horse metaphor, it's like you are getting ready to ride your horse. A pressure wave in the fluid makes the hairs, that are not really hairs, move and send their signal. So the stirrup gets them galloping. Then what? Well, then from the ear through the auditory nerve, through the thalamus, which is the relay station, into the auditory cortex, which is in the temporal lobes, and it goes to both sides of the brain, and not just one corresponding side like the eyeballs do. Here we have the malleus, the incus, and the stapes. Let's talk about pitch perception. Hermann von Helmholtz, he also created the trichromatic theory of the eyes. He came up with the place theory. And then George von Békésy, I guess, I'm not sure that I'm saying his name right, but he found that different pitches cause more vibration in certain places in the basilar membrane. The only thing is we usually hear multiple frequencies at one time, which brings us to the frequency theory. So it is explained as a 500 hertz sound wave will cause hair cells to fire 500 action potentials per second. The downside to this theory is that hair cells can only fire 1,000 action potentials per second, but we hear beyond that range in hertz. Then we have the volley theory, which is the exact same as the frequency theory, but it explains the whole 500 hertz versus 1,000 action potentials. So, in the volley theory, there are teams of hair cells that work together. So if you need 3,000 hair cells firing, what about 3 cells firing at 1,000 each? The duplicity theory is basically a combination of all of these working together. Let's talk about taste. All right, taste is a chemical sense, like smell, and tastes are all interconnected. There are 4 to 5 tastes: bitter, sweet, salty, sour, and umami, which we never had taught to us back in the 70s and 80s when I learned all about the sense of taste. These are not found just in certain places on the tongue like they taught us back in the 70s and 80s, but it's just more concentrated in certain parts of the tongue, and there's not that many in the middle, which makes sense since the food you chew goes off to the sides of your mouth. Taste buds are not the bumps on your tongue, but the pits between the bumps. Those bumps convert the chemicals from the food into neural impulses. They can regenerate. For example, if you burn your tongue and they get damaged, so they get numb for a while, but then you grow new taste buds, so then eventually you're able to taste again. You don't have that numb spot anymore. All right, journey to the brain. The taste bud goes to the neurons that makes 3 major nerves, one in front, one in back, and one from the throat, to the medulla and pons, to the thalamus, to the cortex, which mostly goes to somatosensory cortex in the parietal lobe, and some to the limbic system. The yum-oo factor. So, due to learning we like different foods. Likes may depend on what mom was eating when breastfeeding or foods that were given during infancy, hormones, genetic variations, etc. And then here we have a lovely close-up picture of our sense of taste. Handy dandy note card time. On the front side, please write 5 taste sensations. And on the back, salt, sweet, bitter, sour, and umami. All right, moving on, our sense of smell. So our sense of smell is a chemical sense just like our sense of taste, and our noses are actually very sensitive, and it habituates quickly, most closely tied to memory. So remember, in the last PowerPoint, I talked about the amygdala and how it was called the nose brain. And remember, the amygdala is tied to how we process memory. So we don't know exactly why, but we do know it works. And we'll talk about it again in just a little bit. So there is something called the olfactory epithelium, which is a piece of skin lining on the top of the nasal cavity that has the olfactory receptors that convert odors into smell. So we think maybe it's processed by using that lock and key theory that we talked about with the axon bulbs fitting exactly into the correct dendrites for the synaptic process. So here we go again, talking about the amygdala. The idea of the lock and key theory is that certain odors are maybe exciting to certain receptors, so they will more likely fit in those receptors. And the journey is that the odor becomes neural impulses in the receptors, and they travel up the olfactory nerve into the olfactory bulb, which is in the frontal lobe, and then goes to smell cortex and the temporal lobes, cortex, and the limbic system, which remember, that's where we process memories and have emotions. So this is why they think that smell is so closely tied to memory due to the limbic system activity. And then we also have pheromones. Well, cats have specific organs to detect pheromones. A lot of different animals do. Some scientists think that humans might have something like this too. They think that the reason that women's menstrual cycles synchronize has something to do with pheromones. And also they did a t-shirt study where women smelled men's t-shirts and judged pleasantness and sexiness. And then they find that people choose based on DNA differences, hormone levels, etc. All right, note card time. Olfactory epithelium. And on the back side, it's the top of the nasal cavity, which contains olfactory or smell receptors. We're very close to being done, I promise. All right, next is the sense of touch. We have skin layers, and I'm sure that you guys remember, like in 3rd or 4th grade when we learned about epidermis, and we used to go around telling people, your epidermis is showing, and then we expected them to freak out, and we all giggled, and it was great. So the epidermis is the outer layer of your skin with layers of dead skin cells. Dreamy. The dermis is the inner layer that has the most touch receptors. We have about 5 million sensory receptors within the skin. All right, we have multiple kinds of receptors. To include those listed, we know more about touch than any of the other types, however. The journey to the brain consists of pressure on the skin pushes against axons of touch receptors, which changes membrane permeability, which then changes positive and negative in neurons to make them fire. Signal goes to the spinal cord, up to the brain, into the thalamus, and then into somatosensory cortex and limbic system. The brain is where we interpret these signals, and everyone can perceive them differently and react differently. So a hug to one person might be really pleasant, while to the other person it might freak them out. Body in space is how we know where we are and what we're doing. This is also part of our senses. We've got kinesthesis, which is the ability to sense the position of the body parts in space and their relation to one another. There are receptors in joints, muscles, and in the body. Teens are typically clumsier because their limbs grow too fast for their perception of their body to keep up. Think about the phantom limb theory, which is when someone has an amputation, but they swear they can still feel either pain or some sort of sensation in their missing limb. The vestibular sense is your sense of balance. We get this from the fluids in our inner ear, and the gel is like the water in a water bottle. If it gets overstimulated, then you will get motion sickness. So think about a boat constantly rocking and rocking and rocking and rocking. That is some serious overstimulation, and usually it makes people puke quite a bit. Note card time. Vestibular sense. And on the back, you're going to write your sense of balance. So I, for one, have been diagnosed with a couple of different types of vertigo. And vertigo is basically not having a sense of balance. When the signals tell my brain that I'm walking or standing still, they get all confused, and the signals tell me that I am falling out of a tree or I'm stuck on a rocking boat. So a lot of times that results in passing out or falling over or getting motion sickness just for standing still. Good times. All right. Really quick talk about perception. Top down to bottom up. So top down means using what we already know to interpret what we see. This allows us to recognize and fill in gaps. It can cause errors because we will and have different experiences. So we will have and have had different experiences. Two people can see the same thing and interpret it differently. So think about that great picture that's all over social media right now, where it's a picture of a 6 to one person and it's a picture of a 9 to someone else. Also, we see what we expect to see from prior knowledge, which can also be a problem. So bottom up, there's no previous knowledge to use. You're building from scratch. You're taking everything as it comes. And this can be much harder and take longer. Imagine meeting people one time and then seeing them somewhere else. They might seem familiar, but you only have limited experience with them after you built from scratch the first time you met. So a cow, for example, how hard is it to figure out what it is? What if I say it's a cow? Is it easier then? I'm not really sure, but I believe this is a really horrible picture of a cow. All right, on our next note card, we're going to write top-down versus bottom-up processing. And on the back side, top-down uses previous knowledge, bottom-up, no previous knowledge. So it's like abstract art. You have to figure out what you're looking at based on only what you're looking at. Perceptual constancy is when your brain understands that although some things might look different, some rules still apply. Like using the top-down processing, using prior knowledge for what it is currently seeing. This can cause errors like the moon error. We have cues on the horizon for size and none when the moon is in the sky. So it's the same size, but it doesn't always look like it. Other errors are shown on page 110 in your textbook. Your culture and your upbringing can affect your perception too. People who grew up in round homes, less sharp angled housing, they are less likely to fall victim to Mueller-Lyer illusion, for example. We live in a 3D world, but the image on our retina is 2D. So how we perceive depth. Think about a binocular. The brain gets cues from both eyes, which are in slightly different positions. And that they're in different places, also looking in different places that allow the brain to get cues from the different views. Then there's monocular, which is one eye can still give info. Otherwise, we'd be useless if we got lost. We can use relative size, interposition, etc. There's a chart on page 107 that gives you a really great long list there. Here we've got an example in this picture. Distant objects are higher on the horizon, and distant objects seem smaller. Distant objects become less clear, and converging lines show the distance and the depth. All right, the Gestalt approach. When you look at something, the perception is not just a sum of angles, lines, colors, etc., but rather seen as a whole. You don't see parts, you see the thing itself. Some perceptual laws try to explain how our mind organizes the stimuli to make the whole form. Figure ground is when we look at something, we pull something to the front and push others to the back. They call that in filmmaking racking the focus. So depending on what you do, you see different things. Closure is a tendency to see stimulus as closed shape rather than lines. Proximity is when we group stimuli that are close. Similarity, we group stimuli that seem alike. Good continuation, we prefer to perceive stimuli that seem to follow one another as a continuing pattern. So each thing is its own piece, but what if they're not? It's like that top-down thing where you think the guy must have another leg behind the counter. Here's a great picture that shows different types of perception. And here's another one. All right, note card time. Gestalt on the front and on the back. We've got closure, see stimulus as a closed shape, proximity, group stimuli that are close, and similarity, group close objects together. And we did it. We made it. It's not as short as I wanted it to be, but please make sure that you're reading this chapter and paying attention to the charts and the questions at the end of every chapter. Don't forget, homework is due this coming Sunday at 11:59 p.m.